This is the first of 5 videos for class session 2, and in this lecture we'll be talking about how the resting membrane potential and the action potential are generated. I'd like you to write down at least 2 questions about this recorded lecture. If there are any concepts that were unclear, we can discuss them at the beginning of class, and this will be true for all subsequent lectures as well. You are to bring to class questions you have about the recorded lectures. During this lecture and the next lecture, we'll be talking about how the basic electrophysiological properties of neurons arise. We'll be talking first about the properties of ion channels, specifically voltage-gated ion channels. We'll then be talking about how the resting membrane potential of neurons is generated, and this will include a discussion of the Nernst potential and also of the Goldman equation. We'll then talk about how the action potential is generated and how it's initiated at the axon hillock. In the following lecture, we'll be talking about the passive properties of neurons, the so-called electrotonic properties of neurons, including the membrane time constant and the membrane length constant. And this in turn will allow us to understand how action potentials are propagated down axons. We will begin our discussion of the electrical properties of neurons by talking about ion channels. And what we have illustrated in the figure on the right here are ion channels that are embedded in the plasma membrane made up of the lipid bilayer. These 2 layers of lipids right here make up the surface membrane of neurons, separating the outside of a neuron from the inside. And as you can see, ion channels are typically found in one of 2 states, either a closed state where the pore of the channel is closed, or an open state where the channel is gated open, the pore opens, allowing ions to flow between the outside and the inside of the cell. One of the ways of classifying different ion channels is by the gating mechanisms that lead to the opening of the channel pore. And there are 3 types of gating mechanisms in different types of ion channels. There are voltage-gated channels, ligand-gated channels, and mechanically gated or stress-gated channels. In the next several lectures, we'll be concerned primarily with voltage-gated channels, and in these channels there are amino acid residues within the channel protein that sense the voltage between the inside of the cell and the outside of the cell. And when the inside of the cell becomes more positive, this causes a conformational change in the channel protein, leading to the channel pore being gated open. The second type of channel is a ligand-gated channel, and in fact that's what we're illustrating here. This is a ligand-gated channel that is gated open with acetylcholine. This would be found at the neuromuscular junction, at the synapse. And in this channel there are typically 2 binding sites for the ligand, and when these sites are occupied, this causes a conformational change in the channel from a closed state to an open state. The third type of gating mechanism is a mechanical or stress-gated type of channel, and these are typically the types of channels that are found in sensory receptor cells. And these channels, like all channels in fact, are anchored to the cytoskeleton, which is found just beneath the plasma membrane. And when the membrane is stressed or stretched, this mechanical stimulus is transferred via the cytoskeleton to the channel, and this stress causes a conformational change, opening up the channel pore. A second way of classifying channels is by their ion selectivity properties. When a channel pore opens, this allows ions to flow across the channel pore, but only certain types of ions are allowed to flow because of a selectivity filter within the channel pore. Voltage-gated channels typically are selective to just one type of ion, so we find that there's some voltage-gated channels that are permeable only to potassium, others to sodium, and still others to calcium. And in fact, we classify these voltage-gated channels by their permeability properties. So we'll talk about voltage-gated potassium channels, voltage-gated sodium channels, and so on. Ligand-gated channels are less selective in their ions that they're permeable to. A typical ligand-gated channel that's gated by glutamate will be permeable to both potassium and to sodium. This would be an AMPA receptor opened by glutamate. Still other ligand-gated channels are permeable to calcium as well as potassium and sodium. This would be an NMDA receptor, also gated open by glutamate. Other types of ligand-gated channels are selective for anions instead of cations, and typically these are permeable to chloride. One of the characteristics of all ion channels is that when they gate open, they allow a fairly large flux of ions to flow across the plasma membrane, typically 10 to the 6th or even 10 to the 7th ions per second. 10 million ions per second can flow across through the pore of a single ion channel. So this is a very efficient way of moving ions between the outside and the inside of the cell, just simply to gate these ion channels from a closed state to an open state. Much of what we know about the properties of ion channels has been derived from experiments using a technique, the patch clamp technique, that was developed just a few decades ago. And in this technique, one takes a patch electrode, which is a glass micropipette that has a rather large tip of about 1 to 2 microns in diameter, and one presses this pipette against the surface plasma membrane of a neuron. You then apply suction to suck up a small patch of membrane into the tip of the electrode. This suction causes a high resistance seal to form at the tip of the electrode, so one in effect isolates this membrane patch. Now we can then monitor the ion current flow across this patch of membrane by using a technique called the voltage clamp technique. We'll be discussing this technique in a future lecture, but suffice it to say that the voltage clamp technique allows one to measure the ions flowing between the outside of the patch here and the inside of the cell. Now we know that the lipid bilayer is impermeable to ions, so the only way that an ion can flow across this patch of membrane is if there are ion channels that have been isolated in this patch, and in this drawing we see that there's one ion channel, and if this channel is gated open. So in the closed state, there's no way for ions to flow across the membrane, so we'd measure zero current. However, if this channel gates open, then ions can flow between the patch pipette fluid and the inside of the cell through the open pore of the ion channel, and we will measure a certain current. And in this way we can learn about the gating properties of the ion channel and how many ions per second can flow through the channel when it's gated open. What we see in the top portion of this figure is a record from a patch clamp experiment, and in this experiment a single ion channel was trapped within the membrane patch at the tip of the patch electrode. Now if the ion channel is in a closed state, the pore is gated shut, then no ions can flow across the membrane patch, and we record a zero current, and that's indicated here as zero current flowing across the membrane patch. However, if the channel gates open, then we record a negative current shown here, and as long as the channel remains open, we see this constant negative current. If the channel gates close, the current immediately becomes 0 again, and then if once again the channel gates open, we see this negative current. Now the fact that we record a negative current when the channel gates open is a convention of the patch clamp technique, and then we'll talk about this later, but a negative current indicates that positive ions such as sodium are flowing from the outside of the cell through the open pore to the inside of the cell. Now we can learn two very important things about the properties of ion channels based on these patch clamp recordings. The first is that the transition from the closed state to the open state and the open state to the closed state occur almost instantaneously in hundreds of microseconds or even less. A very rapid transition. The second thing we learn is that when the channel gates open, it always opens to the same extent, so that we see the same amplitude of negative current every time the channel is in open state. Now there are several different ways in which the patch clamp technique can be used, and we will discuss these here. And these diagrams illustrate the different modes of the patch clamp technique. So as we have already discussed, when doing a patch clamp recording, one presses the patch clamp electrode against the surface of the neuron and applies suction. This is called the cell attached patch clamp configuration, and as we've already discussed, in this configuration we can measure current flowing through individual channels that are within this membrane patch attached to the tip of the electrode. Now if we apply additional suction to the patch, we can actually break through this patch, and we then obtain a whole cell recording configuration where the inside of the electrode is in contact with the cytoplasm of the cell. And in this way we are actually, if we record the voltage in the electrode, this voltage will be the same as the voltage inside the cell, and this is a way of obtaining a whole cell voltage recording where we can monitor the voltage in the cell. So this is similar to if we had a sharp electrode and then just inserted it into the cell, but there are important differences between a classical sharp electrode recording of the voltage of a cell and the voltage obtained in the whole cell patch recording technique, and we will be discussing the advantages and disadvantages of recording the membrane potential of a neuron using these two techniques. We'll talk about this in class. Now if we first obtain a whole cell recording configuration and then pull the electrode back away from the neuron, this broken membrane patch will be pulled away as well, and then it will reseal, and we end up with an outside-out configuration of a membrane patch. And it's called outside-out because if you look at the membrane patch here and look at this surface of the membrane, you'll see that this surface here was actually on the outside, the extracellular side of the membrane to begin with. It's right here. And so the outside of the membrane is on the outside of the pipette. On the other hand, if we start with the whole cell, with the cell attached patch recording here and then just pull away, we obtain an inside out configuration because this is the inside of the membrane and it's facing outwards. This shows that you can do additional things to this, but we don't really need this. We can just obtain an inside-out configuration recording just by pulling away from the cell. To review, the patch clamp technique can be used in a number of different configurations, and this can tell us different things about the properties of neurons and the channels that are expressed in neurons. So we can use the patch clamp technique in the cell attached patch configuration, inside out or outside out patch configurations. This is where the isolated patch is attached to the tip of the electrode, or we can record in a whole cell configuration where we're recording the voltage or current of the entire cell. As I've already mentioned, we've learned some very important things about the properties of ion channels using the patch clamp technique. One of these is that the transition from a closed state to an open state occurs essentially instantaneously in well less than a half of a millisecond. And we've also learned that for a single channel species, that when the channel is in an open state, a constant number of ions per second will flow across the channel, because it has the open pore has a certain size which is the same in all channels of that specific channel species. We can characterize the size of the channel opening by measuring how much current flows through the channel pore. And using Ohm's law, we can translate this conductance, this current, into a conductance, and conductance is actually the reciprocal of resistance. And so by using Ohm's law, knowing the current flowing across the channel and the voltage across the membrane, one can calculate the resistance, and then from there the conductance. And conductance is expressed in units of Siemens. And typically ion channels, different types of ion channels, will have a conductance in the picosiemen range. That's 10 to the minus 12th Siemens. And different channels can have a conductance ranging from 1 to 200 picosiemens. We will close this portion of the video by posing two questions, and you can read the questions for yourself. This is an exercise that we will be doing in class. So we'll be breaking up into small groups, and you can discuss these two questions with your fellow students, and we'll come up with the answers together. So we will have a number of these exercises that we will be discussing every day in class, and I'd like you to think about these questions before class, and then we'll be discussing them together.